Mabuhay and welcome to another episode of Bio Buddies. In this episode, we're going to be discussing gene interactions. We will explore the concepts through an example by taking into account conditions when two genes interact and what its effects are. You are familiar with this one? Yes, these are dogs, and as you know, they come with different colors, sizes, and features. Haven't you wondered what causes these variations? In this video we will talk about how the colors of dogs are decided at the molecular level. Specifically, we will talk about this mixed breed of Pekingese and Shih Tzu dog named Mooks, and the genetics of how his color came to be. Are you ready to explore the social life of genes? Dog color is decided by a variety of genes. In this video we will talk about 4 of these genes, the E, B, D, and I genes. But before we dive into discussion, we must first be familiarized with a set of conditions of gene interactions that code for dog color. Let us begin with this diagram. As you can see, there are 2 basic pigments of dog color. This is found in the E loci in the gene melanocortin receptor 1 or MC1R. The eumelanin, which codes for black color, is dominant denoted by capital E and blank, and it can either be heterozygous or homozygous dominant for it to be expressed. The other pigment is pheomelanin, which codes for the red color. It is expressed in recessive state and is denoted as 2 small letter e's. The B gene affects only the eumelanin pigment. In the presence of the dominant allele, the black color expressed by the E gene is retained while the homozygous recessive state turns the black pigment into brown. The D gene affects both the eumelanin and pheomelanin pigment with a dilution effect. In the presence of the dominant allele, no dilution takes place, and the phenotype is retained for the E genotype. In its recessive state, it dilutes the pigment provided by the E loci genotype. Lastly, there is the I gene which only affects the pheomelanin pigment. In its homozygous dominant state, it provides an intense red color. In its heterozygous state, the red color is diluted to a lighter shade, and in its homozygous recessive state, the color provided by the pheomelanin gets diluted to light shades of yellow. In our example, as stated earlier, we will use Mooks. You can notice that his coat color is light yellow. Our goal is to identify its genotype, and we will use the diagram presented earlier. One strategy we can apply to this is to look at the output and backtrack to the genotypes. The outputs are the AND boxes. Among these AND boxes, this color here is closer to the color of the dog. If we follow these backwards, we acquire the combination of 2 small letter E and I. Therefore, the genotype of Mooks in terms of dog color is 2 small letter E and I. How did we arrive at this genotype if there is only one trait that is the color which it codes for? Also, how did we arrive at such shade color if the basic colors were red and black? The combination of 2 genes that code for the same trait yields effects that are a combination of both. Think of it this way. Genes can work like a team to achieve a common goal. The group output is dependent on the members' interaction towards one another, where one member can substitute for another if the task favors his skills, or the member who is skilled in a particular field leads the group. We will discuss a few interactions in the diagram and we'll get back to Mooks later.
The first interaction we're going to discuss is the interaction between E and B loci. The interaction between E and B loci is a form of interallelic interaction. Specifically, it is a recessive epistatic interaction, wherein E is epistatic to B because if E produces a homozygous recessive genotype, it masks the effect of B. This means also that B is hypostatic to the E locus. B gene contributes in pigment production where a dominant allele produces black pigment and brown if recessive. The E allele dictates the amount of pigment to be deposited in hairs. This means that a dominant E allele deposits the pigment of B locus in the hairs and also adds in the eumelanin pigment as enhancement. If the E locus is in homozygous recessive form, the pigment produced by B is not deposited in the hairs, and thus the pigment that is expressed is that of small ee, which is red. You can see how the Punnett square on the right yields a non-Mendelian ratio. This is a recessive epistasis ratio. The second interaction is between E and D loci. The genes in the D and E loci are called collaborator genes. Compared to the previous interaction, This one does not have an allele that masks another allele. Instead, they have a combinatory effect. The dilution genotype, denoted as small caps II, which is homozygous recessive, does not mask the effective genes of another locus, but rather it alters the expression. It modifies the intensity of the color provided by the pigment that's either eumelanin or pheomelanin. The effect is additive. You can see in the right that the ratio is Mendelian. However, it's not exactly like a Mendelian ratio. Why is that? In a Mendelian 9:3:1 dihybrid ratio, the ratio focuses on two alleles of the same gene only, whereas collaborator genes focus on two different types of genes. Since the interaction is within one locus or within two alleles of the same gene, then the interaction is intraallelic. Specifically, since it is shown that the heterozygous state of the alleles yields a combination of both the pigment and the dilution, then the interaction that is showcased is incomplete dominance. Now we go back to MUX. We already know that dilution genes present a collaborator gene characteristic. We also know that within the I locus, there is also incomplete dominance. Since I locus affects only the recessive form of E locus, plus the fact that the dilution will exist if the I locus is recessive, we have a ratio on the right that is quite different.
Gene interactions have long been studied in order to understand how it expresses a particular phenotype. Generally, the significance of genetic interactions focuses on how to specifically predict an offspring that deviates from the expected result. In understanding the genetic compositions of dog color, we can anticipate or prevent possible outcomes that could lead to the benefit of the owner. Scientists were able to identify the specific locations of genes on a chromosome that are responsible for the expression of the coat color on dogs. Fortunately, unlike other species, dogs have few color-associated health problems, and it can be avoided through careful breeding. The lack of pigment anywhere on the dog can make the skin much more sensitive to the sun. Pigment is actually necessary for certain parts of the body to function correctly. So, the inability of a gene to produce melanin can cause health-related issues.
All dogs, no matter what color, are genetically viewed as either black, brown, or yellow-red. All other colors are the result of genetic interaction, wherein mutations in two genes produce a phenotype that is surprising in light of each mutation's individual effects. The mixing and matching of genes gives us the many color and pattern combinations we see in dogs today. In addition to genes, the color of the dog often is related to the dog's purpose in life. Dogs used to protect sheep are often selectively bred to be white in order to blend in with the flock. Dogs bred to herd sheep are selectively bred not to be white. This allows the rancher to identify the dog easily among the sheep and snow. I hope you were able to grasp the concepts of gene interactions today. We will discuss other modes of interactions in genetics, but this time in the molecular level. And that will be a topic in the next video. This is BioBuddies reminding you to stay diverse.